what do I mean by amino acid? I mean, it's got an amino group and a carboxylic acid group. So isn't it ironic that it's typically zwitterionic and that carboxylic acid group is really in its carboxylate state? So let's discuss the zwitterionic life of amino acids.
So I'm going to get into like all this technical detail and stuff about what an amino acid technically is. But the basic idea is that when we're talking about amino acids, we're typically talking about them in the context of being the building blocks or like the letters of proteins. So there are 20 different common amino acids, and they have these different properties. They have this generic backbone part, though, that's going to allow them to link together through peptide bonds. And then they also have this unique side chain or R group, which gives them unique properties. And then those properties are going to influence how the protein folds. And this is why different proteins are going to have different properties and different functions, because they have different sequences of amino acids, and those amino acids have those different side chains and different properties. But what we're going to talk about today is the part that makes them all similar. And this is going to be that generic backbone. So this backbone has this alpha carbon, which is kind of like the central hub. Then the part that's actually going to do the linking, you're going to have this amino group and this carboxyl group. And those actually going to use those amino and those carboxyl groups when they link together through these amide or peptide bonds. And so only these groups are only going to be free at the two ends. So your N terminus is going to have a free amino group. We call it the N terminus because of that amino group, and then the C terminus is going to have a free carboxyl group. So these can be positive. This group can be protonated or deprotonated, and this group can be protonated or deprotonated. But in like our bodies and at physiological pH, all of this stuff, you're typically always going to see this in the protonated form. And when it's protonated, it's going to be positively charged. And you're going to see this in the deprotonated form. When it's deprotonated, it's going to be negatively charged. And we'll get way more into all of this. But how this comes into play is that you often see amino acids drawn in this neutral form, which is, so you have like, this is neutral, or in this totally like non-ionic form, where this is going to be deprotonated and thus not charged, and this is going to be protonated and thus charged. And the thing is, this doesn't really exist. And as we'll get into, this is going to be because of the pKa's of these different things, which is a measure of like how greedy they are for actually taking those protons. And so really what's going to happen is that if you go any if you go any higher in the pH, so if you have more protons available, this is going to get protonated. This is going to get protonated. Well, now this is positively charged, and this is still protonated though, because this was already protonated. And so this is going to be charged, positively charged. What if you go the opposite direction? What if you make things more basic? You take away protons. Now this is going to be negatively charged, but this is still going to be, this is going to be deprotonated still. And so this is going to be negatively charged overall. And there's no pH at which you are going to have this non-ionic form. Instead, what you typically see is what we call this zwitterionic form. And this zwitterionic form, a zwitterion is basically where you have a positive charge and a negative charge in the same molecule. And these kind of make the molecule neutral overall, but you still have the positively and negatively charged parts. Now, don't confuse this with polarity. So zwitterions are polar, but not all polar compounds are zwitterionic. Zwitterionic specifically refers to having like fully charged parts. So now we need to, whereas polarity, you can have like partially charged parts like we see with water. And so we need to get into a little where this charge is actually coming from, as well as what really is a proton. So let's get a little deeper into these concepts. So molecules are made up of atoms, so like these individual hydrogens and oxygens and carbons and all of this neat stuff. And these are made up of these smaller parts, these subatomic particles. So you have the nucleus, which has protons and neutrons, and then it's surrounded by this sea of negatively charged electrons. So protons are positively charged and electrons are negatively charged. Neutrons are neutral, and elements can have like different numbers of neutrons, and then we get like nuclear isotopes. Those aren't really going to come into play here. because they're not charged, but just know that that's a thing. And in fact, when we have hydrogen, the hydrogen version that we normally talk about, like the normal hydrogen, it has no neutrons. And so all it has is a proton and an electron. And if it loses one of those electrons, then it's going to be just a proton. And this is why we can talk about protons as like write them at H plus, because that's what a deep electronated, I guess that's not really a term, but that's what just a proton is, a hydrogen atom without an electron, and one that doesn't have any neutrons. So that is what a proton is and why we can write it that way. The reason why like protons can kind of like come and go, it's not, well, actually protons aren't just like floating around free protons. Instead, they latch onto water and you actually get a hydronium ion or H3O plus. But typically, we just, for simplicity's sake, we talk about them as protons, even though they're often going through like a water transfer. But you can have protons transfer because you have that electron. It gives the opportunity to form a covalent bond. So a covalent bond is when atoms share electrons, and they can share a pair of electrons to form a covalent bond, or two pairs to form a double bond. And these are going to be like strong bonds, but in the case of as opposed to just like attractions between like partially charged molecules. But when it comes to bonds to hydrogen, these are fairly weak, especially if the bond is to something that is electronegative. So something that's electronegative is going to be something that hogs electrons. And so an example is oxygen or nitrogen. They're going to be hogging the shared electrons. And this is going to make it hard for that hydrogen to hold on to an electron, because it just has that single proton trying to like hold on tight. And so these hydrogens can come off or like get donated, or they can, molecules can grab onto them. So when it comes off, you get deprotonated, and when it gets grabbed on, you get protonated. And importantly, this is talking about the hydrogen leaving, but leaving with the electron. So keep the molecule that stole the electron. Now the hydrogen is going to leave as just a proton. And when you have these molecules giving electrons, we call those acids. And when molecules take electrons, we call those bases. And in your solution, you're going to have some free-floating protons, which are really going to be hydronium ions. But the basic idea is that because some molecules are giving these up, you're going to have some of them around in the solution. And the more you have in the solution, the more acidic the solution is. And somewhat confusingly, the lower the pH. So it's because you have an inverse scale. And so the more protons you have freely floating around, the more acidic the solution is, and thus the lower the pH. The fewer protons you have, the higher the pH and the more basic or alkaline the solution is. Okay, and so I like to think of it as kind of like a proton cookie jar. And so the fuller the jar is, the more acidic the solution. And the emptier the jar is, the less acidic the solution. And why this comes into play is because the level of the protons in the jar is going to depend on how many acids you have donating protons and how many bases you have taking protons. And whether or not they take a proton or give a proton is going to depend on something called their pKa, and it's going to depend on how full the jar is. So I like to think of it kind of like a cookie monster. And so the more cookies are in the jar, the more acidic the solution, the lower the pH. The fewer cookies are in the jar, the higher the pH and the more basic or alkaline. Okay, so now you think about a cookie monster. If the cookie, depending on how greedy that cookie monster is, it may or may not like want to give up a cookie or take a cookie. So if it has, we need to talk about this idea of a conjugate acid and a conjugate base. So if something, it's not like something ever is an acid or a base, it goes both ways. And so if it gives up a proton, well, so if it acts as an acid, well, now it has the possibility, it can't give up a proton again unless it's like has multiple protonatable groups or deprotonatable groups, but it can take a proton back. And so you have a conjugate acid, gives up a proton and becomes its conjugate base, which can take a proton. And so if you were to go and look at a molecule, you could find it in its conjugate acid or its conjugate base form. And which one you're more likely to find it in is going to depend on how much, like how greedy it is for that proton, as well as how many protons are around. So if you have a stronger acid, so remember an acid is something that donates a proton, it's going to donate a proton even if the jar is fairly full. But if you have something that's is like a weaker acid, well, now it's going to say, okay, well, the jar's fairly full, I don't need to donate one, right? Okay, well, this isn't really how it works. How it works is that if there's more protons available, it's going to be more likely to run into them. If it is like a weaker acid, it's going to have less of the, or if it's like a weaker base, so base is going to be something that takes a proton. Now imagine that you have a really full jar. There's going to be a lot of protons around to like run into and take. And if you are a strong base, you're going to want to take one of those. And so you're going to take one of those even if if the jar is pretty empty. And so even if there aren't that many protons around, because you're more likely to run, even when you do run into them, you're going to hold on to them tighter. Also, if there's more protons around, then you're going to have then you're going to have more chances to run into them. And so even then, a weaker base could still be protonated. So we need to take into account the pH as well as the pKa. And so the pKa is going to be a measure of the greediness. So basically, the pKa is the pH at which half of the molecules are going to be in the conjugated acid form. So half are going to be protonated and half are going to be in the conjugate base form and are going to be deprotonated. So the pKa is the measure of the pH at which this halfway point is. So it kind of tells us where we draw this 50-50 line between having a 50-50 mix of the conjugate acid and the conjugate base. If we're below the pKa, so if we are at a more basic solution, there's not going to be as many protons available. And so it's less likely that it's going to be protonated. And so you're going to see a higher amount of the basic form of the conjugate base because there are fewer protons available to bump into, and then it's less likely to have one. And conversely, if you are at a lower, more acidic pH, there's going to be more protons available, and so you're more likely to run into them, and you're more likely to thus be protonated. And this is going to be true for no matter what acid we're talking about. But what's going to differ is what this pKa value is. So if you have a stronger acid, you're going to have a lower pKa, which means your pKa line is going to be further over here. And if this means that it's more likely to be deprotonated. So even if you have a higher concentration of protons available, it's not holding on to them very well, and so you still have the protonation. Whereas if you had a stronger base, you're going to have a higher pKa, so a stronger base is a weaker acid, or the conjugate acid form is going to be weaker, so it's more likely to be protonated. Another way you can think about this is which is more stable. Is it's more stable, like determining the pKa of the compound is which state is it happier in, and the happier it is in one state, the harder it's going to be to make it change. So the more different the pH is going to have to get. And we can actually look to the pKa relative to the pH to predict the protonation state. And although differences in the pH, like it's just a one fold different, or this is a difference of 1, well, that's actually telling you it's 10 times more protonated or deprotonated. So if you go up in up 1 in the pH above your pKa, well, now you're only going to have like 9% of it protonated as opposed to 50%. And if you go 2, well, now it's 100-fold. So now you only have less than 1%. And that and so basically, if you go a little bit away from the pKa, you rapidly get away from that 50-50 state. Okay, so this was kind of a long sidetrack, but what we're talking about this is relevant because we're talking about the conjugate acid and the conjugate base forms of amino acids. So amino acids have this amino group and they have this carboxylic group. Okay, so this in the protonated form, remember the protonated form, it has the possibility to act as an acid to give up a proton. So this is the conjugate acid form. This, however, has already given up a proton, and so this is a conjugate base form. And so remember, I told you that this is the form that typically exists. And so when we see this form, this is going to be protonated. And so this is going to be in its conjugate acid form, and this is going to be deprotonated. So it's going to be in the conjugate base form. Now, this is a bit of a problem because we typically talk about this as being a carboxylic acid when it's in this protonated form, and then when it's deprotonated, well, now with this conjugate base, we call it a carboxylate. So when it's in this form, it's actually, this part is not a carboxylic acid group, it's a carboxylate group. And so this is just a minor technical detail, except that it has physiological relevance. And when we actually draw it, we should be drawing it in a more physiological relevant state, such as this, and definitely not in a state like this. So this is a little bit better because this could actually happen at high pH, but this is not going to happen. And to tell us why this is not going to happen, we have to look at those pKa values. So this carboxylic acid group, it has a low pKa. So at around a pH of 2, this is going to predominantly be in this protonated state. So a pH of 2, that's really low. And the reason why this is really low, why this is so acidic, is because you get this thing called resonance. So basically you get this electron density is going to be shared in this carboxylate group. So this is really, really happy in this conjugate base form. And so it's going to require a lot of protons, so a really low pH in order to get it to protonate. And then when you have at a high pH, so this group is going to have a pKa of around 10-ish. So this means you have to get it at a really, really high pH, so really basic conditions in order to get it to deprotonate, because it's happy sharing, it's happy with this positive charge. It's okay with that because it likes having that extra proton there. And so this group is going to be happy like this, and this group is going to be happy like this, and you're going to have to take them far from equilibrium in order to get there. If you go to a pH at which one of those would change, well, then the other one is already going to have been there. So if you go above a pH of like 2, this is going to be deprotonated. But you have to go to a pH of around 10 for this to get deprotonated. If you have a pH of 10, well, this is going to be way, way, way deprotonated, because now we're talking about like we're 8 above the pKa. That's not even on this chart. It's crazy. It's like 10 to the 8. So it's really, it's going to be deprotonated. And if we go further this way, well, if we go to a lower pH, so more protons available, well, this is going to be protonated because as we just mentioned, you have to get to a pH of like 10 to get this to deprotonate. So anywhere below that, it's mainly going to be protonated. And then we have to get to a pH of 2 for this to be protonated. So if we're at a low pH, we're going to have both of these groups protonated, and it's going to have a positive charge. If we go too far this way, we're going to have both groups deprotonated, and so we're going to have a negative charge. And so this like idea of this non-ionic form, although it's nice to draw, and it's typically drawn this way by convention, this is not actually like a thing. So when you see something like this means that the carboxylic acid group is actually, so when we see it in like its real form, this carboxylic acid group is actually in its carboxylate form, which is the conjugate base form. But to get there, it acted as an acid. And to get here, so that this is going to be the conjugate acid form, it already acted as a base in order to get there. So this idea, it's just some terminology things, as well as like thinking about how this is actually existing in a solution in a more realistic way. And this can have consequences because these groups can then be more reactive and things like that. Plus, zwitterion is just a really, really cool word, and it makes me happy. And I like that word. So remember, zwitterion is something that has a positive charge and a negative charge, but they cancel each other out, and so the molecule is neutral overall. And this is different from polarity, although this is a polar molecule because it has separation of charges. Polarity does not necessarily imply that you have full charges, whereas zwitterionic does. And so if you see ion, that means like a charged molecule. And like a fully charged molecule, because you actually have like an imbalance of electrons and protons in that like area of the molecule or whatever, in that bonds, that sort of thing. And so you're going to have fully charged parts, but neutral overall. So remember that your carboxylic acid group already acted as an acid. So now it's in its carboxylate form, and your amine group already acted as a base to get here. So in the form you find them, they're typically like opposite of what you would think based on what, like we say by like amino acid, we talk about a carboxylate group. So at least one end will always be ionized. The neutral form is often drawn by convention, but doesn't really exist. At low pH, both of these will be protonated and you'll have a positive charge. At a high pH, both ends will be deprotonated and you'll have a negative charge. So I hope that helps you better appreciate the zwitterionic life of amino acids. The final note is that some amino acids have a third pKa. This is sometimes called like pK3 or pKr, and this is because they have an ionizable side group. And so these are going to be like our basic and our acidic amino acids. And when we talk about them in this form, we're typically talking about, we call the acidic ones, the ones that act as an acid, and we typically then see them in their conjugate base form. So similarly to how we talk about like aspartic acid is typically in its aspartate form, glutamic acid is typically in its glutamate form. Then for the positive side, we have like glycine and arginine, and these are going to have already acted as a base and typically be protonated when we see them. Okay, hope that helped. I also want to mention that the exact pKa's are going to vary from amino acid to amino acid. So that's what on like exact values on here. They're going to be slightly different depending on what this R group is because of things too complicated to talk about here. But basically, this is like drawing more electrons away or not, and various things like that. This pKa can be slightly different. And also remember that if you have a protonated side group or deprotonated side group, if this group can be charged, then you might have a fully charged amino acid, even if the backbone is in this zwitterionic state. So then the molecule overall would not be zwitterionic. Okay, so hope that helped.